Hello everyone, and welcome to today's live broadcast, Refinement of Epigenetic Approaches in Neuroscience, presented by Dr. Richard Lee. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Today's event is sponsored by Agilent Technologies. Agilent Technologies, Inc. is a global leader in life sciences, diagnostics, and applied chemical markets. It's a premier laboratory partner for a better world. Agilent works with customers in more than 100 countries, providing instruments, software, services, and consumables for the entire laboratory workflow. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want and at any time you want during the presentation. Just click on the green Q&A button located in the lower left of your presentation window and type your questions into the box that appears on the screen. We'll answer as many questions as we can, as far as we can at the end of the presentation. Also, please notice that we will be viewing the presentation in the slide window. To enlarge your window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you are having a problem. This is an educational webinar, and thus offers continuing education credits. After the webinar is over, please click on the CE button located on the bottom left-hand corner of your webpage and follow the process for obtaining your credits. I would now like to introduce today's speaker, Richard Lee, PhD Assistant Professor from Johns Hopkins University School of Medicine. Richard Lee is a Johns Hopkins University lifer. He received his BS in Biomedical Engineering and a PhD in Molecular Biology and Genetics at the Johns Hopkins University. Currently, he is an Assistant Professor in the Department of Psychiatry, and his main research interest is elucidating the epigenetic mechanisms of the HPA axis and the neuroendocrine of hormones in the context of psychiatric disorders. He employs both candidate loci and genome-wide approaches to identify neuronal genes and pathways that are epigenetically affected by stress and glucocorticoids. He has designed and implemented both microarray and sequencing-based platforms for genome-wide DNA methylation projects, including the CHARM platform and the SureSelect MEthylSeq target enrichment system for the rat genome. Please join me in welcoming Dr. Richard Lee. I will now turn the presentation over to him.
Thank you for the wonderful introduction. So as mentioned, I will talk to you today a little bit about epigenetic approaches in neuroscience. It's a bit of a work in progress because, my training is originally in epigenetics, and as I've started to dive, into the realm of psychiatry and neuroscience, I realized that the methods that I normally use were somewhat inappropriate or inadequate. So I had to kind of pick up things along the way, different tools and techniques to be able to effectively examine epigenetic modifications in the brain. So today, I'll just give you in the beginning, I'll give you a brief, a crash course background on epigenetics, and then tell you a little bit about some of the tools that we use. And then, because I'm not just describing techniques and platforms, I'm going to tell you a little bit of my research background and how it evolved over the past couple of years and necessitated for me to think a little bit about neuroscience and how I could merge the fields of neuroscience and epigenetics to answer some of the questions. And then I will give you some justification for a genome-wide platform called MEthylSeq for looking at DNA methylation, and then go into detail about the Agilent SureSelect mouse MEthylSeq platform and how I've used it to answer some questions that I had about glucocorticoid signaling and epigenetics. And then as I think a little bit more about discoveries in neuroscience and in the brain, how I had to pick up some neuroscience techniques along the way to answer some of those questions in a more appropriate context. So briefly, what is epigenetics? The textbook definition is, of course, the study of heritable changes in gene function or phenotypes caused by mechanisms other than changes in DNA sequence. It's basically a study and measurement of transcriptional context or potential and inertia. So it's the architecture within the nucleus that you would kind of examine to see whether a particular gene is able to transcribe or make mRNA, or it becomes silenced. So there are three kind of flavors of epigenetic changes, epigenetic modifications. One that I will talk primarily about today is DNA methylation. The other one being histone modification, and the third one, which is fairly hot topic now these days, is the non-coding RNAs, such as microRNAs and link RNAs, or long non-coding RNAs. To tell you briefly about DNA methylation, it's a CH3 modification that gets covalently attached to the base pair cytosine, and not in all cases, but generally you could think of it as methylation being associated with gene silencing. So on the DNA itself, on the base pairs of DNA, especially at the CpG dinucleotide, the C, or the cytosine, becomes methylated, and what happens with that is that it confers binding activity to specific proteins, such as MECP2, that binds methylated CpGs and then silences transcription. On the other hand, if it's unmethylated, it's thought to just promote transcription because there's nothing that is hindering transcription factors from binding. And I will talk a little bit about the interaction between methylation and histone modifications in the next couple of slides. And of course, we have the histone modifications. These are proteins in a octamer format that the DNA is wrapped around. And there are multiple modifications, such as acetylation, phosphorylation, and even methylation on different histone residues that promote either gene transcription, gene activation, or silencing. So this is involved in euchromatinization or heterochromatinization of histones. And this is another slide just demonstrating how complex the histone modifications are. And this is one of the reasons why I tend to shy away from working on histones. I do have some experience working with histones. I quickly realized it's just quite complicated to think of which modifications lead to what and what proteins bind these different modifications. And then, of course, you have not only the histone modifications in its variety, but different enzymes that, as I mentioned, that modify the histones. In this case, we have for example, HDACs that are histone deacetylase inhibitors that remove acetyl groups from specific lysine residues on histones, and so it promotes heterochromatinization of your histones. And then oppositely, there's the histone acetyltransferases such as P300 that provides or attaches acetyl groups to these lysines to promote gene transcription. And then, of course, we have the untranslated RNAs that are not made into proteins, but they perform very important functions in terms of gene regulation. So in a sequence-specific way, they bind their, for example, mRNA target, and they affect transcription translation of specific genes. And as you may already know, there's plenty of role that epigenetics plays in biology and disease. One of the prime example of that being X inactivation. We know that females have 2 X chromosomes, one of which is inactivated. So when the one of the chromosomes are inactivated, it actually uses all three mechanisms of epigenetics to achieve that purpose. And then we have cell differentiation, especially when you think of stem cells. Any cells in your body, they have the same DNA sequence, yet they perform very different functions. And part of that is being mediated by epigenetic differences. And then, of course, you have cancer. Obviously, there are mutations, genetic mutations that occur in cancer, but there's a lot of epigenetic mechanisms as well. Since turning a normal tissue into tumors requires cellular reprogramming through epigenetic modifications. And of course, there's genomic imprinting, which basically, based on whether you get your chromosome from your mother or your father, there is differential gene expression. So coming back to X inactivation, it's a very good example of epigenetics because the inactive X chromosome actually employs all three mechanisms. So you have HP1, which is the heterochromatin protein 1, binding to one side of the chromosome, and then the other side is covered by exist RNA, which is the X inactivation silencing transcript, which is a long non-coding RNA that coats the inactive X chromosome. And then when you look at the figure on the bottom, you can see that largely the inactive chromosome, the X chromosome, has lots of methylation content. So if you look at the gene PGK1, which is on the X chromosome, in males versus females, you'll see that for males that need that one X chromosome copy, most of the genes are unmethylated, whereas in females, the methylation is anywhere from 40 to 50% to indicate that at least one of the two X chromosomes are methylated. And this is genomic imprinting. This is the classic imprinted locus 11p15.5. You have the IGF2 and H19 genes, and the pink color basically shows, pink boxes show the genes that are transcribed off of the maternal chromosome, whereas the blue boxes show genes that are paternally transcribed. And so this is a perfect case where epigenetics plays a role, and the mechanism that has been established, that has been demonstrated, is that the differential methylation of regions that are called DMRs, or differentially methylated regions, confer binding to insulator proteins such as CTCF and nucleophosmin that block the access of enhancers to specific genes. In this case, if it's methylated at the bottom panel, there is no binding to an insulator protein, so the E representing an enhancer could activate the IGF2 locus, whereas on the maternal chromosome there is an insulator protein and it inhibits, prevents access of the enhancer to the IGF2 locus so that it kind of activates the closer H19 gene. So you get this reciprocal expression pattern when you have genomic imprinting, and it occurs, of course, through epigenetic mechanisms. So the tools of epigenetics, I will briefly mention several of these. Of course, there's detection of DNA methylation, histone modifications. These are, as I will go into detail on some of the examples, the kind of bread and butter of epigenetics. And then we could do either candidate gene approaches or genome-wide. This is now has become much more, I guess, comprehensive due to the sequencing platforms that are out there. And then one of the tools that I will go into greater detail is the fluorescence activated cell sorting. So this is not detecting epigenetic modification per se, but I think that it's a very important tool for you to be able to enrich for specific populations of cells. And this becomes highly relevant when I talk about approaches in neuroscience towards the end of this talk. So I will talk primarily on about DNA methylation, because that's that is a bulk of my research. And how we detect DNA methylation, there's quite a few tools you could use, methylation-sensitive enzymes, but I choose bisulfite conversion of DNA because I think it is the most, it provides the best base pair resolution in terms of detecting methylation changes. So what happens during bisulfite conversion of DNA is that the cytosine becomes sulfonated. This is, you buy a nice little kit from QIAGEN or Zymo Research to convert your genomic DNA. And so if the cytosine in the context of CpG dinucleotide is methylated, it is prevented from becoming sulfonated, whereas if it is unmethylated, it becomes sulfonated. And doing a hydrolytic deamination step with desulfonation, the unmethylated cytosine becomes uracil. And so in the subsequent PCR procedures, it becomes a T. Whereas if the cytosine is methylated, it survives the kind of the bisulfite conversion step and it remains a C. So what happens at the end is you have a bisulfite converted sequence where all the C's that are not in the CpG context have been converted away to T's, and then you have C's only remaining in the CpG context where the C was methylated previously. So when you compare your final sequence to the original sequence, you could tell whether a piece of DNA was methylated before the bisulfite conversion. And this is a kind of a very simple schematic of how we determine percent methylation. Previously, what we did was we bisulfite treated the DNA, did PCR amplification, and then we took it to the core, or if you have a ABI 3100 sequencer, you did Sanger sequencing. So then what you did is to have primers on either side, go sequence over the CpGs and determine whether there are what the CG composition is at that particular dinucleotide, and then what you wanted to do, if you wanted to be a little bit more specific, is to clone it into a vector using topo cloning kit, and then pick a bacteria colony that represents one type of species of DNA, and then when you did Sanger sequencing on that particular plasmid with that DNA sequence, you could either get a C or a T to indicate methylation status. So then for a particular experiment, to determine whether a CpG is methylated or not, you would pick like 20 colonies and look at the ratio of CGs versus TGs. And then the method that was developed, I guess, in the past 10 years or so is a method called bisulfite pyrosequencing. This is where you get a PCR product, and using the PyroMark MD system, assay basically assess the percent methylation at CGs versus TGs at hundreds of millions of PCR products. And so this is the idea behind pyrosequencing. So you have a PCR product that you want to assay at the CGs for methylation, and then you have a pyrosequencing primer, and then with incorporation of either TG or CG, basically you have pyrophosphates that are released from the incorporation of the nucleotide. And so that's where the name pyrosequencing comes from. And what that pyrophosphate does is it drives, an enzyme called sulfurylase drives the formation of ATP, and the ATP is being used by the luciferase enzyme to generate light output. Basically, it's a luminometric assay that you're detecting, and it's the gold standard for candidate gene assessment of DNA methylation, and it's highly quantitative compared to counting colonies. And I mentioned these two different methods because it turns out that the genome-wide sequencing platform is a kind of a comprehensive way of colony sequencing. And so then, I prefer to then validate some of these genome-wide hits with pyrosequencing as an independent method of validation. So there's also the detection of histone modifications. You could actually cross-link and sonicate histones. So what you get is basically the histone octamers with the associated DNA wrapped around them. And then you use specific antibodies that recognize specific modifications of histones to pull down pieces of chromatin, and then you reverse the cross-link, and then you PCR amplify across your region of interest. And now that's the candidate gene approach. And now, you know, with microarrays and especially with next-generation sequencing, you could actually look at all areas across the genome that were associated with that particular histone modification. And you could also do this with antibodies against specific transcription factors that were cross-linked to your DNA, and then do what's called a ChIP-seq, chromatin immunoprecipitation followed by sequencing. So this is a, you know, the next two slides show you some papers that were published looking, implementing some genome-wide methylation platform. So this is the CHARM platform developed by Dr. Andrew Feinberg here at the Epigenetics Center, and I was involved in building the rat genome version. And then I've also been involved with looking at hydroxymethylcytosine in the context of drug addiction. And so this was using a technique called MEDIP, or HMEDIP, antibody against hydroxymethylated cytosines, and then throwing basically the associated pull-down DNA on the sequencing machine. So there are candidate gene approaches that you could do, and then there are genome-wide methods to detect different epigenetic modifications. And let me just add that I didn't mention too much about microRNAs, but this is very similar to, you know, obviously RNA-seq, that you could get these RNA species and then make libraries out of them and then put them on the either microarray platform, or in this more commonly used next generation sequencer to detect the very different types of RNAs, such as long non-coding RNAs and microRNAs. Now let me switch gears to what I've been working on for the past couple of years, and why my work, some of the things that I did in the past couple of years really created a need for me to move into, you know, some of the techniques and approaches in neuroscience. So I've been involved in looking at the epigenetics of stress, hormone exposure, or glucocorticoids and stress, and its impact on the brain. And what I was able to find in the past couple of years was that glucocorticoids can actually directly alter DNA methylation. And as obviously this is work being done by other fantastic labs, you know, around the world. And it turns out that these methylation changes persist. And that's one of the reasons that really got me into looking at stress hormones in the context of psychiatric disorders. So let me briefly talk to you about the stress response. The stress response is mediated by the HPA axis, consisting of the hypothalamus, the pituitary, and the adrenal glands, with endocrine outputs from each one of these organs that are necessary to mount the stress response, your fight or flight response. And of course, the end result is the release of stress hormone cortisol from the adrenal glands. And I guess arguably I would say that the acute stress is actually quite good for you, because it mobilizes your energy and makes you alert. Your heart rate goes up, your blood pressure goes up, and like when you exercise, in the short term, I think it's quite good for you. But unfortunately, a lot of us are living at this day and age are exposed to a lot of chronic stress. Sitting in front of the computer like what I'm doing now, and working away on grants and things like that. It creates a lot of stress, day after day, and week after week, and so forth. And unfortunately, that exposure to chronic stress leads to a host of diseases, such as cardiovascular disease, hypertension, obesity, diabetes, and of course, of particular interest to me are psychiatric diseases such as depression, anxiety disorders. And in the case where, the stressor is very high in magnitude and I guess acute, you could also be afflicted with PTSD. So there's quite a bit of literature linking stress and stress hormone exposure to psychiatric diseases. And as I mentioned, it's a risk factor for anxiety and mood disorders, as well as neurological diseases such as Alzheimer's disease. It turns out that stress plays a pretty big role in this particular disease as well. And one of the reasons that really got me excited about looking at stress and glucocorticoids in psychiatric diseases is that unlike, there's a lot of things I don't know about bipolar disorder and depression, but at least if stress plays a big role in these diseases, what's attractive about stress hormones is that you could actually measure it in the blood. And as I mentioned, there are plenty of literature linking glucocorticoids in psychiatry. To go into greater detail, it turns out that hypercortisolemia and glucocorticoid resistance is often comorbid with depression. And I also found out a couple of years ago, reading through one of these little books on depression, that it turns out that glucocorticoids, or human stress hormone cortisol, are one of the few molecules that can actually trigger mania and depression in bipolar disorder. And I've also had the opportunity to work on Cushing's disease. So this is where you have a tumor in the pituitary or the adrenal glands, and it basically results in the excess release of cortisol. So they have hypercortisolemia. And it turns out that about 60 to 80, 90% of these patients develop depression. And what's also interesting is that when the hypercortisolemia is resolved through surgery, removal of these offending tumors, a lot of the cases, the depressive symptoms are alleviated. So there's a kind of a nice cause-effect relationship between glucocorticoids and psychiatric disorders. This is a very large epidemiological study looking at hundreds of thousands of people that were treated with glucocorticoids for non-psychiatric symptoms such as asthma and lupus, rheumatoid arthritis, and things like that. And it turns out that people that were exposed had much higher incidence of depression, mania, panic disorder, and suicidal behavior. So I'm just kind of hammering in this, notion that stress hormone actually plays a huge role in psychiatric disorders. And this allowed me to kind of think a little bit about the role of genes that regulate the HPA axis or the stress response, and to see whether they're actually involved in psychiatric disorders. And it turns out that many of these genes that are either target genes of glucocorticoid signaling or can actually regulate glucocorticoid signaling in the HPA axis are actually involved with psychiatric disorders. So these are numerous studies that have linked these genes to psychiatric disorders, and this allowed me to formulate kind of an idea as to how epigenetics actually plays a role in mediating exposure to stress and stress hormones to psychiatric diseases. So you have, everybody has their homeostatic cortisol dynamics, that obey circadian rhythm and, exposure to stressors and so on. So you have this kind of normal level of homeostatic level of plasma cortisol. And then what happens when you're exposed to prolonged exposure to cortisol or stress is that it starts to alter DNA methylation and other epigenetic modifications of these HPA axis genes. And once they alter epigenetics, they start altering their function. And when that happens, they start modifying the homeostatic cortisol dynamics. And how that's important is that these modifications and changes in gene function of HPA axis genes actually cause alterations of some of the target genes in the brain, such as BDNF. This is the brain-derived neurotrophic factor, and TH, which I'll talk about in a little bit, which is the tyrosine hydroxylase, which regulates dopamine synthesis. So you have prolonged exposure to stress affecting epigenetic modifications of HPA axis genes, where some of these HPA axis genes are actually very important in neuronal function. And so then the model that I have is that these ultimately lead to persistent changes of these genes, of course, and then to psychiatric disorders. Let me just mention briefly how cortisol works. It's a hydrophobic molecule based on cholesterol, and so it enters the nucleus of the plasma membrane quite readily. It binds to the glucocorticoid receptor. Glucocorticoid receptor dissociates, enters the nucleus, and it acts as a DNA binding transcription factor. And so then you have target genes of glucocorticoid signaling, as I mentioned to you before, the tyrosine hydroxylase and the BDNF genes that are either silenced or turned on. And I will focus on one particular target gene of glucocorticoid signaling called FKBP5. I just kind of passed through rather quickly, but the FKBP5 gene is actually a very important gene in regulating glucocorticoid signaling, and it turns out it's also a target gene. So I got really interested in FKBP5 because some of the SNPs or genetic variations in this gene were associated with depression, and from our group with showing that it was associated with bipolar disorder, and other people have shown that genetic variants in this gene were associated with PTSD and alcoholism and so forth. So it seems like the FKBP5 was a very interesting gene in the context of glucocorticoid signaling, but also in psychiatric disorders. Mentioned to you, showed you this slide before. Let me just add here that FKBP5 not only regulates the intracellular glucocorticoid signaling, but it actually is a target gene. And so what that does is that FKBP5 gene, once it becomes transcribed from glucocorticoid signaling, goes to the cytoplasm and acts as a ultra-fast negative feedback protein to limit additional glucocorticoid signaling. So as you could imagine, with excess cortisol levels and excess transcription of the FKBP5 gene, you could get this negative feedback loop where additional glucocorticoid signaling is prevented. So I wanted to see, given these findings, whether chronic exposure to stress hormone and glucocorticoids leads to epigenetic changes in FKBP5. So I did this really kind of simple, very simple experiment of providing glucocorticoids or corticosterone. So the rodent version is corticosterone. I'm sorry, the rodent glucocorticoid is corticosterone. So we gave them in the drinking water for 4 weeks, and then after 4 weeks, we tested them, for anxiety-like behavior on the elevated plus maze, and and the tail suspension test, and so forth. And then after the test, we collected, and examined, their brain regions for any changes in, transcription and epigenetic changes. And then we had the other half of the cohort to see, and then we let them go for another additional 4 weeks in the absence of any additional corticosterone to see whether any of these changes, if they were there, would persist in the absence of glucocorticoid signaling. So this was a kind of a initial candidate gene study that we did, and it took a very long time for me to actually find where DNA methylation was changing in the blood. And what you see here in figure B is a particular CPG in intron 1 So the y-axis is percent DNA methylation using determined using Pyro sequencing, and then the x-axis are the number of weeks. So at 4 weeks, we treated them with glucocorticoids, corticosterone, and then in the second half of the 4 weeks, we let them recover. And you can see here in figures B and C that there were at least 2 CpGs that underwent epigenetic changes, and they lasted for about a week in the blood, and then they kind of returned back to baseline by the time the animals were sacrificed at the 8-week period. And then when you look at the expression of this FKBP5 gene, you can see that after 4 weeks, it becomes elevated, and then during the recovery period, it comes down to baseline. So this was in the blood. And then we show in the brain that there's another region. So it's a very kind of tissue-specific epigenetic change that's occurring with glucocorticoid treatment. You can see here at the top panel, which is the hippocampus, and the bottom is the hypothalamus. And you can see in panel figure B that there's a kind of, at 4 CpGs, there are methylation changes that occur with glucocorticoid treatment in another intronic region. And what's interesting about this study was that in the 8-week period, when they've gone without glucocorticoids for 4 weeks, you can see that the methylation changes persist. Now, I'm showing you these figures to emphasize the fact that the effect size, or the percent methylation difference, is quite small. And this was when I was just starting out learning about glucocorticoids and doing these DNA methylation assays. And this kind of got me to think, is this a neuron-specific or a non-neuron-specific event, and what kind of circuitries are involved, and so forth. And that kind of got me started with looking at specific neuronal populations in other experiments. And of course, to show that, demonstrate that cellular heterogeneity plays a part, what I used here in this particular experiment is a cell line. It's a very simple mouse hippocampal cell line, and of course there's no cellular heterogeneity there because these are neurons that were transformed, and so it's fairly homogeneous in tissue culture. And what I was able to show is that, sure enough, when you actually look at one population of cells, the methylation differences are so much bigger when you're not looking at bulk tissues. And then, so figure A shows the expression changes with glucocorticoid treatment of the FKBP5 gene, and then figure B shows basically that one intronic region in the mouse brain where we saw methylation change, and sure enough, there's a fairly big methylation change that also persists in the recovery period. Now, we treated these animals these cells for about 7 days instead of 4 weeks, and we were able to observe a fairly robust epigenetic change. And of course, did some experiments just to show why is the glucocorticoid methylation change important. And these two figures kind of show you why they are. So, I got these cells again that were previously treated with glucocorticoids, and then when you treat them again with an analog glucocorticoid dexamethasone, you can see here on the top figure that when it's been previously demethylated by previous exposure to glucocorticoids or dex, the next time you treat the cells with glucocorticoids, the response is higher. So methylation is playing a role in mediating a more robust expression of this FKBP5 gene. And then to make sure that the intronic region has functional relevance, what I did was then clone that piece of DNA into a CpG-less vector. So this is a commercially available vector or plasmid that all the CpGs have been changed to, I guess, TGs, so that the plasmid itself does not get methylated. And then you clone in your fragment of interest, and then you compare when that fragment has been in vitro methylated or when it's unmethylated, so that the piece of DNA is driving the expression of a luciferase gene in a methylation-sensitive way. And you can see here in the left panel, on the left side is the FKBP5 intronic region. So you can see a fairly robust expression when the methylation is not added to those CPGs compared to the one all the way to the left, where it's been heavily methylated by an enzyme called SSS1 methylase. And then on the right panel, you can see that a consensus glucocorticoid response element that binds to the glucocorticoid receptor also shows similar methylation-sensitive activity. So at least from these figures, it seems like as far as FKBP5 is concerned, that it has this methylation-sensitive, that it responds epigenetically to chronic glucocorticoid treatment. So then I started asking the question, what other genes are out there that change epigenetically with chronic exposure to stressor glucocorticoids? And then, of course, if you do find multiple targets, are there any common pathways that we could think about? So this is one of the, the beautiful things about doing genome-wide investigations, because you're not only looking at one gene, now you're kind of generalizing your finding to the genome, and you're able to now identify, trends or even pathways or gene sets that might be important in disease. So let me tell you briefly about the SureSelect platform. The one that I use is a SureSelect platform by Agilent. Basically, you're shearing DNA and attaching adapters. You're ligating adapters to your sheared DNA, and then you're using the Agilent technology, where they've developed specific RNA baits against specific genomic loci, and you're pulling these sequences out of solution. And then after enrichment using streptavidin-coated magnetic beads, so these RNA beads, the RNA species are biotinylated, and then you use streptavidin-coated beads to pull them down out of solution with a magnet. And then you bisulfite treat the DNA, and then you put it on the sequencing machine. So it sounds quite simple. The experiment itself is quite simple, but the analysis is actually quite challenging. So then let me just tell you briefly about what this platform entails. So this is the mouse methylome target platform, and it targets the CPG islands, tissue-specific DMRs, or differentially methylated regions, and it takes into consideration or targets multiple regulatory features such as CPG shores and shelves, DNase I hypersensitive sites that we know have been shown to be associated with binding of transcription factors, histone modifications, and so on. So it's got a lot of these regions across the genome, and I prefer for now using such platforms rather than whole genome sequencing, because for lots of reasons it's still quite expensive, and these are very gene-focused platforms. And what we do after sequencing is a whole slew of bioinformatic tools that we implement. So this was done by our bioinformatics group led by Dr. Peter Zandi and Fayez Saifuddin. There's we align the sequences, perform quality control, and then there's a whole slew of statistics that get generated in terms of what the target percentage is, and the read depth and coverage and so forth. And then there are methylation calls. So basically, with the aligned sequences at a particular CPG, you're determining how many reads represent that particular CPG, and then using those reads, percent methylation is determined. And this is the figure that I showed you before, just to emphasize the fact that when you do a genome-wide sequencing, it's a fancy way of doing colony sequencing. So you're looking at, basically when you do genome-wide sequencing, each read represents kind of a bacterial colony that you would have done if you had done colony sequencing. And you use this method to basically, in a semi-quantitative way, calculate or assess percent methylation. So this is a experiment that I did with the glucocorticoid treated animals. So I used one cohort to look at the FKBP5 gene, and then once I had some findings regarding epigenetic, with epigenetic changes at the FKBP5 locus, I then looked at genome-wide using the methyl-seq kit. And so these are some of the statistics that we that are provided by the the bioinformatic analysis. So you get the total paired and reads, how many of them are mapping, the mapping efficiency, and then we look at how many number of CPGs are being covered, and then how many of them are on target, meaning that how many of them are actually targeted by the methyl-seq platform. And then from there, we calculate an average of, for each CPG, or on average, what's the read depth for each of the CPGs that are being targeted. And in this case, we got about anywhere from 24 to 31x coverage, which is fairly decent. You probably don't want to go below like 10x coverage for these types of assays. And so we, for this particular experiment, we pulled our DNA samples with, I guess, 2 animals representing 1 sample. And then this is the kind of the output that you get at the end. We use a analytical algorithm called BSmooth or BSseq, and basically for sets of there's a window of CPGs that are changing in methylation. So then what the algorithm does is kind of smooth over those areas, and then we look for regions where, from the smoothing between the cases and controls, or treated or untreated, or vehicle-treated animals, there's a statistically significant difference. So what the algorithm does is it provides lists of genes, of course the genomic coordinates and their associated genes and all the statistics for those regions, and it ranks them based on, I guess, a arbitrary statistical parameter. And it also provides a very nice PDF plot of your samples and what the methylation patterns are. In this one, at the bottom is a PDF plot that shows the regions, a particular DMR, differentially methylated region between vehicle and corticosterone-treated samples, blood samples in these mice. And then what you do then is, for the hippocampus at the top and for the blood at the bottom, you then, design specific candidate assays. And I mentioned to you the bisulfite pyrosequencing platform in the previous slides. You go in and try to validate these specific regions. And you can see here that for both the hippocampus and the blood, you can see a nice independent validation by bisulfite pyrosequencing. And then what you can do with genome-wide approach is to look at specific pathways. And I performed the KEGG pathway analysis in both blood and the hippocampus. And what's really interesting is that, there's lots of these very disease-relevant pathways that pop up from the analysis that are relevant to, diabetes and obesity and psychiatric disorders. I showed you in the previous slide in that nice figure with the effects of chronic stress, that these are actually these pathways are actually relevant to some of these diseases that are associated with chronic stress exposure. And then you could also do some interesting analysis, such as, where are these DMRs? Where are these differentially methylated regions? And what we found is that a lot of them are in the intronic regions. So this is very consistent with other studies of stress and glucocorticoid signaling, where they show that the glucocorticoid response elements, where the glucocorticoid receptor binds to DNA, is actually in intronic regions. So this is very consistent with those studies. And then when you look at the regions in the context of CPG islands, we originally thought that CPG islands were these regulatory regions and so forth, but a lot of these regions, as far as glucocorticoid signaling is concerned, fall outside of CPG islands, which I thought was interesting. But of course, this is something that's been published for quite some time in cancer as well. And then one of the things that I thought was kind of interesting was that it turns out that glucocorticoids and the genes that are epigenetically modified by them, remarkably, between in blood and the hippocampus, there's a very similar percentage of loss of DNA methylation events. So what we know from the action of glucocorticoids is that they could upregulate or downregulate genes, and in this case, as far as epigenetics is concerned, that most of these events are loss of methylation events. So I'm not sure how that reconciles with genes that are silenced by methylation. And of course, finally, we, having provided you with the background in epigenetics and some of my research, now we go into some of the epigenetic approaches that I've adopted and implemented in neuroscience. So this is kind of work in progress, and some of the things that I've been thinking about, and some of them are implemented and other ones I would like to do. And so let me just say that one of the primary functions of neurons, of course, involves action potential propagation, with ion channels and receptors, and of course neurotransmitters. And so why study epigenetics? I think that epigenetics could be very complementary to neuroscience, because it allows us to examine long-term gene dysregulation in the brain. So you have neuronal differentiation, neurodevelopment, especially sexual maturation of neurons, during adolescence and so on. And then there's exposure to toxicants such as lead, BPA, alcohol, and then other environmental factors such as stress, famine. And of course, we know that epigenetics plays a role in psychiatric and neurological disorders, which I think, I hope that, neuroscience will shed tremendous amount of light on. And I think that all of these fields will, all of these topics will require a bit of epigenetic approach to further augment neuroscience platforms and techniques. So the main obstacles that I encountered when I started thinking a little bit more about epigenetics and the brain is that there's a tremendous amount of tissue and cellular heterogeneity in the brain, and of course it's the most complex organ in the human body. And when I say cellular heterogeneity, I tend to, I meant, neurons versus, the glia, the microglia, and so forth. But also within the populations of neurons, there's, even in a same brain region, let's say the hypothalamus, there are specific neurons that perform very diverse functions, and so presumably have different epigenetic signatures. So that is something that I had to think about a little bit. And then, of course, when you want to do, really nice epigenetic experiments, you want to enrich for these, these neuronal populations, or even from neurons, specific ones with specific function. But then at the end, if you, do the sorting techniques and so forth, how much do you have at the end? So the limiting tissue amount was a huge problem for me. So let me just tell you, as I mentioned before, about fluorescence-activated cell sorting, I think it has become an indispensable tool for me in terms of isolating specific neuronal types for epigenetic experiments. So basically, this involves, in one set of experiments, labeling your nuclei that you've isolated, in this case from human postmortem tissues, with a neuron-specific antibody, in this case NOI-N or FOX3. And then what you do then is, the prepared nuclei from neuronal and non-neuronal cells, feed it through the cell sorter, and then there's a excitation wavelength, and then there's the emission of fluorescent molecules that are attached to the antibody against these specific neuron-specific proteins, and then the machine is able to physically sort antibody positive versus antibody negative or antibody negative populations of cells. And then what I did then is to look at a particular glia-specific gene. There's a very conserved region in the promoter of GFAP, or gliofibrillary acidic protein, and it's highly expressed in non-neuronal cells, and so you see that there's a huge methylation difference between the NOI-N plus versus the NOI-N minus populations of cells. You could also do this, obviously, with blood. I think this is what was the original intent of the FACS sorting machine, right? Because they use it for immunophenotyping or for immunology. So what I did in this case is to label T cells, B cells, and neutrophils with their specific CD receptor binding antibodies, and then sort, and then to show that actually the methylation changes in the blood do not occur in as much in B cells or neutrophils, but actually they occur in T cells. So this is a method that lots of now epigeneticists employ in neuroscience. And so when I started to look at my animal model using this approach, so on the left figure, you can see that there's a, once again, in the mouse cortex in this case, fairly significant methylation difference between the neuron positive versus non-neuronal cell populations at the GFAP promoter. And then the middle panel shows a kind of the bulk cortex tissue that that I use for methylation analysis, and you can see that there are methylation differences, but it's very tiny, and it's only significant in one CPG. And then what I did then is to FACS sort these tissues, as I did in panel A, and then when you look at the same FKBP5 region in the NOI-N positive glucocorticoid versus vehicle-treated animals, that there's a much bigger effect size with statistically significant differences. So this kind of demonstrates that the importance of cellular homogenization, meaning that you're for doing epigenetic research in neuroscience, and this is just the beginning, that you'll need to kind of enrich for either the neuronal fractions or a very specific population of cells. So this is a work by a colleague and a collaborator of mine, and this is actually probably one of the best methods that I've seen, better methods to isolate specific population of cells. So this was work done by Dr. Minae Niwa in our department, and this was published in Science in 2013. And what she did in this experiment was basically use a fluorescent retrograde bead or beads with different colors, excitation or emission wavelength, and injected these retrograde beads into the frontal cortex versus the nucleus accumbens, as indicated by green versus red. And what you see here is that what happens here is that over time, about 3 weeks, the beads retrograde to the ventral tegmental area, which is the hub for dopaminergic neurons. So you're basically labeling dopaminergic neurons based on their different projection sites to the nucleus accumbens or the frontal cortex. And during that time, what they do is that they've stressed these animals, and these animals, of course, it gets further complicated into a genetic disease, psychiatric disease model versus the wild type. And what they noticed is that in these animals, transgenic animals that have the human transgene for a gene called DISC1, disrupted in schizophrenia 1, when they're stressed, their dopamine levels go way down. So then what they did was then, isolate the ventral tegmental area after the stress regimen, and then trypsinize the cells, and then they FACS sorted it based on the different wavelengths from the green versus the red. And what they show is that extracting DNA from these frontal cortex specific dopaminergic neurons, the promoter actually is hypermethylated in these transgenic animals that are stressed. So the E is for environment, or ones that are stressed, and GXE is the gene environment interaction group, where the animals, the transgenic animals, were exposed to stress. And then when they do, so in this case, they're looking at colony sequencing. Each row represents, number of CPGs that were assayed. In this case, it's 11. And then the black represents methylated, black box represents methylated, white box represents unmethylated. And you can see here that there's fairly dense methylation patterns in these transgenic animals that were stressed. So there's an interaction between the gene, the transgene itself, and exposure to stress. So this was a really cool experiment. And then I, in a subsequent publication, collaborated with her to show that first to replicate this colony sequencing experiment using pyrosequencing at the tyrosine hydroxylase gene. The patterns are a little bit different, but you could still see the methylation changes that occur. And then also showed in other genes such as BDNF and FKBP5 that there are other genes that are changing epigenetically. So this was another even better method to even go deeper into specific types of neurons that, so these are neurons of the ventral tegmental area that are both dopaminergic, but yet based on their different projection sites, Dr. Neal was able to isolate these two populations and do epigenetic analysis. And to finally to add that there were no methylation changes in the ones that the neurons that were projecting to the nucleus accumbens. And then, of course, there are other methods that you could use. In this case, you could use genetic models. So you could have labeled dopaminergic neurons in other ways using crossing, animals that carry the Cre recombinase enzyme driven by a dopaminergic neuron promoter, and then you cross them with floxed tomato gene-carrying mice to label dopaminergic neurons, and then you could combine that with like a retrograde bead to specifically now label dopaminergic neurons only. So there's genetic models that you could implement. And then, of course, at the end of these procedures where you're enriching for specific neuronal subtypes or even neurons to do your epigenetic assays, what do you do when you have so little amount of DNA? So then this is a challenge that I had to think about, and I actually used a small animal study to demonstrate that you could actually get decent sequencing libraries from very small amounts of starting material. So I used a animal model of Prader-Willi syndrome. I know it's kind of a bit of a gear change shift here, but it's a animal model that carries a SNORD116 deletion. So this is a particular SNP RNA cluster. And as I mentioned to you about genomic imprinting, this is a genomically imprinted loci where the paternal deletion leads to hyperphagia. So these animals or humans eat a lot. It's uncontrolled eating, and then they develop obesity. And so we used this animal model, and we looked at the hypothalamic arcuate. So this is a region of the hypothalamus that controls, feeding and satiety and so forth. And what I did was to use use needle biopsy. So this is a very small area of the hypothalamus, and I could only, get a tiny little punch using a blunted needle to get probably a few milligrams of tissue. And from this amount of tissue, I was able to generate both RNA and DNA. And so before I jump into epigenetics of this particular brain region, I wanted to see is there even a rationale to look at epigenetics in the first place? So using the RNA extracted from these samples, I performed RNA-seq, and then found that there's a whole slew of these genes that are implicated, that are differentially expressed between control and the Prader-Willi syndrome model. That justified looking at epigenetics to see what is mechanistically driving these expression changes. So this is a work that's ongoing right now, but we've successfully sequenced basically libraries made from about 50 to about 150 nanograms of starting material. So this is similar to the amount of DNA that you would get from FACS-sorted experiments. So the animal work that was done by Dr. Niwa, it ranged anywhere from about 200 to like 50 nanograms of DNA after you FACS-sort a particular dopaminergic neuron, a species of neurons. And so instead of using the traditional methyl-seq platform or the protocol, which requires 3.5 micrograms of DNA for making libraries, we use a TruSeq DNA methylation kit by Illumina, and then combine that with the mouse methyl-seq by Agilent. So this basically platform uses target enrichment first, and then bisulfite conversion before you do the adapter ligation. So in this particular step, you tend to lose a lot less DNA, and so you can actually afford to start with much less material. So this is the starting material that we had. So it's about, it ranges from about 150 nanograms to about 50 nanograms of DNA. And you can see here from this tiny little hump, this is DNA that's been sheared at about 176 base pair average, that we didn't have much to start with. But then when you go through this particular procedure to the end, you could actually get a fairly decent amount of sequenceable library. So this is, you notice that the size is bigger to indicate that the adapters have been successfully ligated, and this is a high sensitivity screen tape on the tape station, which is the next generation bioanalyzer, to basically show you the size and the quantity of your library. And then, still a little bit skeptical about how good these libraries are. I don't want to spend thousands of dollars at the sequencing core to sequence these samples if there's nothing in the middle. So then I performed a MiSeq experiment. So this will cost you about $1,000 at the core to perform it. Then this is not to do percent methylation analysis, but just to see whether there is alignable sequences from your library. And so far the libraries look pretty good, considering that we started from a very small amount of material. So then it remains to be seen. After this result, I will be putting the samples on the HiSeq, the much larger platform, to do the methylation analysis, and hopefully find DMRs specific to the animal model, and then validate by pyrosequencing. So these are the kind of the epigenetic approaches that I've shown you that need to be kind of modified specifically for tackling the challenges in neuroscience. And now I'd like to acknowledge the people in the psychiatry department, including Dr. Niwa and others, and in endocrinology, my longtime collaborator Gary Wand, and of course the people wonderful people at Agilent, Carolina, Jonathan, and Josh, who've been tremendously helpful in implementing the methyl-seq platform. Thank you.
Thank you for that informative presentation, Dr. Lee. It's time for Q&A. If you have any questions you'd like to ask Dr. Lee, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your questions into the box that appear on the screen, and click on the send button. We'll answer as many questions as the time for available. If we are unable to answer your questions, Dr. Lee will follow up via email following the presentation. So, our first question is, why do you have to validate genome-wide sequencing results by another method?
Yeah, thank you for this question. I would love to just be able to do these genome-wide assays and then do the pathway analysis and so forth and call it a day, but we've had lots of experiences where we've done the genome-wide assays, but then they don't validate. So this was especially a problem with some of the array-based platforms that we had used. And if you're looking at you're looking at different tissues or cancer versus normal, I mean these are big differences that you could actually robustly validate using pyrosequencing. But then when you're working with glucocorticoids, and especially in tissues that perform similar functions, like dopaminergic neurons and things like that, methylation differences might not be all that big, in which case some of the platforms might not be as good for detecting them. I mean, they will give you genes and regions for you, saying that this might be important, but unless you validate them, it's really hard to know whether that's a real finding or something that the algorithm, an analytical algorithm that just kind of spits out at the end of the analysis.
Thank you so much. Another audience member has another question. Is it possible to do chip assays such as ChIP-seq on neuronal isolates?
Yeah, that's a very good question. I've stuck mainly to DNA methylation because I think it's fairly convenient. It has been convenient for me because when you deal with postmortem DNA, postmortem tissues, I think for a lot of the samples the mRNA is toast. So I'm not sure what kind of results you could get from mRNA, RNA-seq, and then you look at histone modifications, and also once again some of the proteins might have been degraded as well. And so, DNA is one of these very stable, or DNA methylation is one of these very stable modifications that you could actually detect in tissues such as, from postmortem studies. And another thing is, you don't need as much starting material. And so, I've done some chip assays, and, the reproducibility, I mean, so don't get me wrong, you could definitely do ChIP-seq on small number of cells or small sample tissue samples, but I think at least from my experience, the reproducibility is a lot lower than when I work with DNA. So because of that reason, and of course with all the whole million gazillion modifications that's found on histones, I kind of shy away from chromatin like ChIP-seq or chip assays. Yep, thank you.
There are wonderful questions coming in, but we only have time for just one more. Dr. Lee, an audience member would like to ask, why do you use target enrichment? Wouldn't it be better to do whole genome sequencing?
Yes, so this is often asked, I guess, especially like NIH grant reviews and things like that. I know obviously it doesn't get any better. You don't get more comprehensiveness than doing whole genome bisulfite sequencing, because you're getting the entire 3 billion base pairs of DNA from that. But it turns out that I mean, what do you do when you get all that information? It's an analytical nightmare, I think, and also because there's lots of repetitive sequences that make up more than 50% of the genome, and so and this is also demonstrated or emphasized by people who've studied whole genome approaches, and it turns out that like good 80% of CpG methylation from whole genome assays do not provide much useful information. Genes I think make up about 1.5 to 2%, I think, of the genome, and so finding robust methylation differences in gene deserts, I'm not sure what you could do with them at this point, unless you could link them to some sort of a long non-coding RNA or something like that. So I like using the target enrichment because it's very gene-centric, and it targets also regulatory regions that may change in methylation and has functional relevance. So I like to focus on those. And of course, there's the economics, the financial aspect of it. If you're going to do or sequence the whole genome bisulfite sequencing, you're going to have to sequence basically a whole lane of HiSeq. So that's about $2,300 to $2,500 of one lane for the entire genome of one sample, and that's going to provide you at best about 10x coverage. Whereas if I'm doing the target enrichment, as you've seen, you could multiplex. In the example that I showed you, I loaded 4 samples per lane, and then they give me about 25 to 30x coverage, which I think is a lot better than the 10x coverage all over the place that you would get. So there's definitely an economic cost here, savings that you would get from doing target enrichment as well.
Thank you once again, Dr. Lee, for a wonderful presentation, and thank you, Agilent Technologies, for making today's educational webinar possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing for up to June 16th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who have missed today's wonderful live event. That's all for now. Thank you for joining us. We hope you come back again. Goodbye.